Yeah, thank you so much for the introduction. Hi, everyone. Once again, I'm Cindy Lee from the National Cancer Institute, United States. Very excited to be the first speaker of the session today and present my work, Fair Molecular Feature Selection Unveils Universally Tumor Lineage Informative Methylation Sites in Colorectal Cancer. I'd apologize for not being able to attend this wonderful conference in person and would like to thank the organizers for allowing me to give this talk virtually from the other side of the Atlantic Ocean. Before I dive into the work, let me first briefly introduce the motivation behind it. One of the long-term focuses of my colleagues and me is to understand tumor heterogeneity and evolution. In each individual, the tumors harbor somatic alterations and evolve into diverse tumor cell subpopulations. Across individuals, it is widely understood that there are commonalities and distinctions in the tumor evolution processes and the associated molecular changes. A good grasp of these commonalities and distinctions among cancer patients is crucial to the development of accurate cancer diagnostics, prognostics, and potentially more effective treatment strategies. Previously, my colleagues and I have used epigenetic changes like CpG methylation to trace tumor evolution in individuals. CpG methylations could behave differently when it comes to status inheritance. Many are more transient and have high turnover rates, making them uninformative for tracing tumor lineage, whereas some others are more persistently inherited during cell replications, which makes them highly informative. Leveraging this characteristic of CpG methylation, my colleagues and I managed to build a method called Scooter, which is published in Genome Research in 2023, and we use it to understand the CpG methylation dynamics in a diverse colorectal cancer patient cohort with single-cell methylation sequencing data. Scooter iteratively teases out CpG methylation changes uninformative for tumor lineage reconstruction to infer a stable tumor lineage for each patient. In the meantime, based on these tumor lineage tree structures, we were also able to simultaneously evaluate the tumor lineage informativeness of the CpG sites for each patient. In other words, we managed to evaluate how stably inherited along the tumor lineage each CpG site is within each patient. So after successfully using Scooter to characterize the CpG methylation changes along tumor lineages in each individual, we now want to ask a question on a population level. Is the dynamics of CpG site methylation universal among all cancer patients? In other words, for a CpG site that is tumor lineage informative in patient A, is it also tumor lineage informative in patient B and C, etc.? So this is actually a pretty tricky question to answer because to understand the universal behaviors of molecular features like CpG sites in the entire patient cohort, we first need to ask, how exactly do we fairly choose a set of features to represent the entire cohort? And before I proceed, I'd like to clarify that in the remainder of the talk, I will use the term feature and site interchangeably. In the context of single-cell CpG methylation, for example, ideally, we want to compare all patients on the same sites across all cells. And in this way, we don't have to worry anything about fairness, right? We can simply just use all of the sites sequenced to represent the entire cohort. But in reality, we're inevitably given sparse cell-by-site single-cell recoverage matrices for individual patients. And by coverage, I mean the number of reads sequenced. When a site is covered, it has at least one read in a given site in a given cell. In single-cell methylation sequencing, we'll observe some patients with a larger number of well-covered CpG sites, which are have at least one read across many cells, and some with a smaller number of such sites. We cannot simply combine all of the CpG sites covered in each patient to represent the entire cohort, as the poorly covered sites introduce noise and cannot be further used to understand the universal CpG methylation dynamics. Alternatively, if we naively analyze only the intersection of best covered sites of all patients, we are left with very few sites. This makes it necessary to develop an approach so that we can choose a subset of well-covered sites that can represent the entire patient cohort in a balanced manner, while not losing too much information as a result of limiting our analysis to only a small subset of the best covered sites. In other words, we would like to select the maximum number of sites that enable apple-to-apple comparisons across patients. To this end, we introduce the molecular feature selection problem, which aims to choose features that fairly represent an entire cohort. In this problem, from a given collection C of sets, each set Ci for a given patient I is composed of elements Cij, each of which correspond to a distinct molecular feature. To select a rich and balanced set of molecular features, one needs to come up with the largest subset of molecular features such that not only each feature is present in at least a user-defined number of patients, but also each patient contributes to the set of select features in a balanced manner. We have proven that this problem is NP-hard. I would also like to highlight that the molecular feature selection problem that we define here is very different from, although related to, the well-known hidden cell problem, which achieves a very different goal. The hidden cell problem asks to identify a minimal set that hits or intersects with each of the sets in the collection, as opposed to finding the largest subset of elements to represent the cohort as described in our molecular feature selection problem. To systematically solve the molecular feature selection problem, we introduce FALAFEL, which stands for Fair Multi-Sample Feature Selection. The primary goal of FALAFEL is to provide a fast and reliable solution for the fair selection of molecular features across different patient samples to represent the entire patient cohort. When finding the optimal solution for feature selection, FALAFEL not only achieves balance across all patients, but also ensures that each patient receives sufficient representation. I also want to clarify that despite the fact that this work is motivated by our previous research involving single-cell methylation sequencing, and I'll for sure discuss the application of FALAFEL also in the same context, FALAFEL is designed for more general purposes and is potentially applicable for other molecular features like single nucleotide polymorphism, gene expression, etc. Now, allow me to walk you through the workflow of FALAFEL. The input of FALAFEL is a collection of sparse binarized cell-by-feature single-cell sequencing depth matrices for individual patients. The matrices for the patients can be combined into a single patient-by-feature matrix S, in which entry Sij represents the fraction of cells with coverage in patient I on feature J. To simplify the problem, we binarize matrix S into matrix S bar using a user-defined coverage threshold P. If Sij is larger or equal to P, Sij becomes 1, otherwise it becomes 0. We also further remove sites well covered in two-field patients. A site well covered in less than user-defined K patients is dropped. We then use this combinatorial optimization approach called integer linear programming, or ILP for short, to select an optimal set of molecular features with S bar as input. The goal is to have an output in which each feature shared by at least user-defined K patients, and each patient harboring at least a user-defined fraction Q of all selected molecular features. Now let me give you a more detailed view of our ILP formulation. Given the pre-processed binary input matrix S of size n times n prime, where n prime denotes the number of features not eliminated in the pre-processing steps. Given a user-defined threshold Q, which denotes that for each patient, the minimum proportion of feature Sj chosen by the formulation for which Sij equals 1. Let R be a binary vector where Rj equals 1 indicates that feature J is chosen by the formulation. Then our formulation chooses features with the objective to maximize the number of well-covered features chosen, while subject to the constraint that each patient must have at least fraction Q of well-covered features in the entire set of selected features. We use the famous ILP solver Gurobi to solve for this problem. The systematically chosen features output by our ILP-based optimization can then be used for downstream analysis. And now let me show you how FALAFEL performs on simulated data. To evaluate the scalability of FALAFEL on different numbers of patients in the input, we generate random binary matrices for 10 to 50 patients, respectively, each with 1 million sites. For each patient count, 3 random instances were generated. From this box plot, we can see that the runtime of falafel is reasonably increasing as the number of patients grows. This result indicates that falafel is capable of scaling to input with larger numbers of patients. And now let's take a look at how falafel works on real data. We also apply falafel to the same colorectal cancer patient cohort I have previously mentioned. As we can see, the patients in the single-cell methylation dataset have very uneven numbers of cells, and each cell have different numbers of CpG sites sequenced. Due to the sparsity issue commonly seen in single-cell sequencing, many CpG sites have different coverage across patients, and we can use falafel to systematically select a subset of CpG sites to represent the entire patient cohort. So the next thing I want to show you in our application to real data is that falafel performs fast and robust feature selection. In this particular application, we're looking at two cases. One is the entire colorectal cancer cohort of 9 patients, and the other is the subcohort consisting of 4 patients that all have primary tumor sites in left colon. Despite the large input size, falafel was able to generate results within 60 seconds on a single CPU for both cases. Notice that there could also be more than one optimal solution for the problem. To assess whether these solutions contain the same set of sites, I also shuffled the order of columns in the input matrix for the left colon subcohort. Falafel picks almost the same features every time, with an average similarity of 99.9%. Moving on, we compare falafel selected sites against naively chosen sites that are well covered in a high fraction of patients. In the left colorectal cancer cohort, the naively chosen sites are heavily biased towards patient CRC11, whereas among falafel sites, that bias is reduced. The result clearly shows that falafel reduces bias against or towards individual patients. We have the same observation in the entire cohort as well. The naively selected sites are heavily biased against 2 patients and towards 1 patient. And again, among the falafel chosen sites, the bias are reduced. Notice that there are 2 patient samples that are very underrepresented because they have a worse coverage than others, and they actually happen to be 2 of the 3 patients with primary sites in the right colon. In this particular case, if we ignore that underrepresentation when choosing the features, this creates biases against right colon cancer patients. So this is certainly one of those scenarios that calls for the use of falafel. We also show that falafel helps discover cohort-wise universal behavior in CpG methylation changes. In our previous work scooter, we have introduced a Jensen-Shannon distance-based metric called lineage-informativeness to characterize the CpG methylation dynamics. For the purpose of this talk, you just need to know that the metric is a number between 0 and 1, and the larger it is, the more informative the corresponding CpG site is for tumor lineage reconstruction, and the more stably inherited and epimutation-like the CpG site is. So here on the left, we show that the lineage informativeness of falafel-chosen CpG sites for the left colon subcohort is highly correlated among all patient pairs. And as for the deviation of the lineage informativeness of the sites among all patients, and we see that in both the left colon subcohort and the entire cohort, most of the falafel-chosen sites have very consistent behaviors across all patients. We further show that falafel is able to better identify CpG sites that are universally tumor lineage informative. Compared to randomly selecting CpG sites for the entire left colon colorectal cancer cohort, falafel selected sites demonstrate a much higher level of correlation in lineage informativeness across all patient pairs, which once again highlights that a balanced set of molecular features needs to be selected before reliable cohort-wise comparisons can be made. We also want to highlight that falafel helps discover some interesting biological insights. For both cohorts, we categorize falafel-chosen sites based on their lineage informative level. Here, the blue dots and bars represent universally uninformative sites, and yellow represent universally informative sites. In both cohorts, universally lineage informative sites are enriched in the inter-CpG island region, whereas universally uninformative sites are enriched in a CpG island. This suggests that CpG methylation changes in colorectal cancer progression is non-stochastic in terms of genomic locations. Perhaps this is not surprising at all because CpG islands are typically functionally important, and methylation changes in CpG islands may be selected against during tumor evolution. So in summary, we present falafel, a principled fair molecular feature selection scheme that introduces algorithmic fairness in comparative patient profile analysis. We apply falafel to the problem of fair selection of CpG sites to a colorectal cancer patient dataset, and our results demonstrate that falafel rapidly and robustly identifies a maximal set of CpG sites that are well represented across the entire patient cohort. We show that falafel ensures fair representation of molecular features across all patients while uncovering cohort-wise universal behaviors of methylation changes. Our investigation into the falafel selected sites revealed interesting enrichment of universally lineage-informative CpG sites in the inter-CpG island regions, which indicates a degree of non-stochasticity of methylation changes in colorectal cancer progression. These findings demonstrate the ability of falafel to identify lineage-informative sites and provide valuable insights into the underlying biological mechanisms of cancer progression. We would like to apply falafel to more diverse patient cohorts and further explore its effectiveness and generalizability in clinical settings. We also would like to use falafel to aid the design of diagnostic and prognostic biomarker panels with fairness in mind. So I would like to thank my mentors, co-authors, and the entire tumor evolution and genomics sections of the Cancer Data Science Lab at the National Cancer Institute. Without them, this work would not have been possible. Thank you so much for your attention. And I will take any questions.
Thank you for your great presentation. Are there any questions?
Yeah, so
please use the microphone.
Yeah, so I'm curious, when you're talking about levels of informativeness, is this being done with all data in essentially in one data set, or are you splitting this into a training set and then assessing it on a set of data that was not used for training the model to identify those?
Thank you so much for the question. So yeah, unfortunately, due to time constraint, I was not able to explain lineage informativeness in its full context. But essentially, this lineage informativeness talks about how well a CpG site is inherited, given a tumor lineage structure. So it's basically a tree. Right, and in every patient, we were able to establish that lineage structure for every single patient. And to evaluate the lineage informativeness of a CpG site, we just look at how stably inherited it is along the tree in that patient. So there is not really anything as training set or anything like that. It's simply, that we build a tree and then we evaluate this metric using that tree, and that is and that CpG methylation value is from the single cell methylation sequencing in individual patients.
We would have time for one quick question. Nobody? Okay, then let's thank our speaker again.